How do I get to my calendar? Before we begin, could I ask somebody to click on the link to their calendar and then click on, come on, brother's taking his time, and then click on the link for the micro lecture in the calendar and let me know if that link works. Could somebody please do that now? Mm. All right, for those of you who are adding now, I'm showing you my calendar. Can you go to your calendar and click on the micro lecture link and find out if that link works? It looks slightly different than the link I sent you.
I tried to do that and it did not connect into this lecture.
Thank you. All right, that means I have to delete all of the links. How do I do that? I didn't create these links. I'm not sure where they came from. I knew that two of them were really different because I don't give them names like Biology 260. But the other one was close to the name I named our course. So I was wondering if that one was working. And unfortunately, I can't delete them all at once. I've got to delete them one at a time. And that means the links and the calendar, wherever they came from, do not work. So you should not use the links you got from the calendar. You should get the links that I emailed to you. Any questions about any of that? All right. Before we begin, are there any other questions? Okay, I'm not hearing anything. I'll leave that up and I'll get more of those later. No, I'll leave that for the lab. So today is April 10th, and we should be continuing chapter 1 and chapter 10 in the lecture. Do let me remind you that you have a few things due this week. This Saturday, you have the worksheet for chapter 1 due. And then on Friday at 8 a.m., you have lab 00 and lab 01 that are due. You also have the environmental quiz that is due 11:59 p.m. this Saturday. 11:59 p.m. on Saturday is the normal time for the worksheets, both the lab and the lecture being due. But obviously, I have to have your lab 00 and lab 01 in beforehand because at 5 p.m. or something like that on Friday, I have to turn in a list of the students who are attending the class and who's not. And then those who are not will be dropped from the class. And how I'm going to do that is by taking attendance by lab 00 and lab 01. Any question about any of that? All right. My voice is still a little bit off from my cold, so please bear with me. I might have to talk a little bit more quietly, in which case I'll try and turn up the microphone. Of course, I don't know how to adjust the microphone. So you'll have to turn up your volume. Let's go to chapter 1. Okay, I told you where microbes can be found essentially everywhere on the planet Earth. The exception would be obviously where there's lava or magma. Microbes are numerically the most abundant life forms on the planet Earth. How many microorganisms are there? We're talking about prokaryote cells. You should realize the numbers I give you. If we're talking about all microbes, including viruses, they would be about 10 times the number that I'm going to give you. So I'm only talking about prokaryotic cells, meaning bacteria and archaea. How many microorganisms are there? In 1 gram of soil, there's about 1 times 10 to the eighth microbes in that gram of soil. This will depend on how dry the soil is and how much organic matter is in it. There are more organic matter that's in it, and the wetter the soil, it'll be a little higher than this number. So this would be your, I don't know, your regular garden soil, although our garden soil at the moment in Western Washington, Western Oregon is pretty wet. So maybe there'd be a little more microbes. But around 1 times 10 to the eighth microbes. Now, when you're in the desert soil, very dry, very little organic matter, it'll be less than this. But
Excuse
this is
me?
our average soil. What?
I don't think you're sharing your screen.
Ah, thank you. Oh, how do I get that? There, am I sharing my screen now?
Yes.
All right, thank you. Whenever I'm doing something like that, let me know and I will try and make the corrections. So there are about 1 times 10 to the eighth microbes in 1 gram of soil. 1 gram of soil is about 1 milliliter of ocean water, meaning it's about 1 gram of ocean water. Do you think you'll have more or less microbes in the ocean water? This is either yes or no question, people. You got a 50% chance of getting it right.
Less.
Yeah, you're right. There's less microbes in a gram of ocean water or 1 millimeter of ocean water. The oceans are considered a desert on the planet Earth, meaning they have less life forms and less nutrients than most other places on the planet. Now, the exception is obviously the desert, like Sahara Desert. That would be about comparable to normal ocean water. And in 1 gram of human feces, do you think you'll have more microbes than 1 gram of soil, or do you think you'll have fewer? It's a
More.
50-50, yeah. 50-50 chance, people. Don't be afraid to guess. The worst you're going to be is wrong, and we're just going to tell you, no, that's not correct. You want to take a guess again? But yeah, you have more microbes in a gram of human feces, especially if it's a fresh human feces, than you do in soil. On one human being, you have more microbes than in soil. And of course, on the entire planet Earth, you have a lot more microbes than 1 gram of soil. The numbers are shown in this slide here. So for 1 gram of soil, we have about 1 times 10 to the eighth. Depending on how many organic matter is in that gram of soil and how much moisture is in it, like I said, if you're in the desert, you have closer to 1 times 10 to the sixth microbes in the gram of soil. In ocean water, we have about 1 times 10 to the sixth microbes in 1 milliliter or 1 gram of ocean water. 1 gram of human feces, if it's fresh human feces, you have about 1 times 10 to the 11th microbes in a fresh gram of human feces. However, if somebody were to go to the bathroom in the desert and then it were to dry out, you would only have about 1 times 10 to the sixth microbes in that gram of human feces. So once again, it depends on the weight of the feces, how much water is in the feces, how much organic matter is in the human feces, mainly water, and how fresh the feces is. So straight out of the person, you're going to get around 1 times 10 to the 11th microbes per human feces. And that'll be about 55% of the wet weight of the human feces. So when you go to the toilet, just think about it. 55% of the weight of that human feces is microbes. On one human, we have about 3.8 times 10 to the 13th microbes. That's on one human or in one human. On the entire planet Earth, we have around 1 nonillion. That is one with 30 zeros after it. You've probably never heard of a nonillion before. And for comparison, the total human population is a little less than 8 billion, or 8 times 9 zeros after it. Any question about any of that? The point of all this is microbes have astounding numbers, essentially wherever we look. When we're talking about microorganisms, you should know that microorganisms may cause disease, at least certain of them. And if the microbe can cause disease, we call it a pathogenic microbe. You should realize that most microorganisms do not cause disease. Only a small percentage of all microbes are pathogenic or disease causing. Any questions about any of that? So most species of microorganisms do not cause disease. When we're talking about disease, it's important to distinguish between an infectious disease and an inherited disease. An infectious disease is caused by a microbe or a worm. Give me some examples of an infectious disease.
Flu.
The flu. It's a good
The
example. Anyone else?
A cold.
Yeah, cold. Those are all infectious diseases.
COVID.
Say again?
COVID.
Yeah, COVID. That's an infectious disease. You're naming all viral diseases, but we could have a bacterial infection like Staphylococcus aureus, skin infection. That also is a infectious disease. That differs an infectious disease from an inherited disease. An inherited disease is caused by someone's genetics, meaning they'll be born with an inherited disease. And what's an example of an inherited disease?
Certain cancers.
Possibly, if the cancer runs in the family, then it would be an inherited disease. Not all cancers are inherited diseases, so you should make a distinction there. What's another example of an inherited disease?
Cystic fibrosis, CF.
Oh, cystic fibrosis. As long as it runs in the family, which most do. There's a few that might be like a spontaneous mutation. Another one would be sickle cell anemia. an inherited disease, which the child will be born with. Somebody gonna make another example? All right, here we're looking at some microorganisms. They are small. They're microscopic, obviously. There's some microbes. That's a fungus. There's a microbe that actually looks like a white blood cell to me. And there's some microbes there. And here, actually, these red things are um, HIV virus coming out of a T4 cell. They come in various shapes and sizes. Let me blow this slide up and you can see the sizes. So right there, this is about 1 micrometer, a little bit, oops, too far. A little bit longer than 1 micrometer, but just a little bit. And then the length of it or the width of it is less than 1 micrometer. Here, these are 10 micrometers, so these are a little longer, bigger cells. And then that there, I can't see that. 10 micrometers, so this one's about 50 to 70 micrometers long. And then this is 50 micrometers here, which is smaller than this, but that's not actually a cell. Each of these green dots is a cell. This is a colonial microorganism. And actually, you see these dots here? This is the parent colony. And this parent colony has 6 baby colonies inside of it. The parent colony will open up to release the babies, and the baby colonies will go elsewhere and colonize other areas. And then there's 50 micrometers there. So this cell is bigger than 50 micrometers. But these microorganisms, which are not exactly an organism, they're virus, they're much smaller than 50 micrometers. So just giving you about the size. A micron is about the size we see under a light microscope. Smaller than a micron, you'd have trouble seeing it under a light microscope. I'm not sure if you can see Volvox with your naked eye. You might be able to see the parent colony with your naked eye, but most of the other things you would not be able to see with your naked eye. All right, microorganisms play beneficial roles on the planet Earth. They play beneficial roles for humans. They decompose organic waste. That's very important because when something dies, if the microorganisms and the fungi did not decompose it, the waste would just build up on the planet and the nutrients in it would never be recycled. Because microorganisms and fungi decompose organic waste, it makes these nutrients available for other organisms in the ecosystem. Microorganisms are important because they are producers in the ecosystem. A producer is something that can fix carbon from carbon in the atmosphere, making generally sugar, which is important for all living life forms. Even if you only eat beef, you get that from eating the cow. And where does the cow get its food? From eating the grass. The grass is a photosynthetic organism. So the grass is a producer. You can have some microbes which are producers, but they're not photosynthetic. They engage in chemosynthesis to make sugar. And we'll talk a little bit more about that later. Microorganisms are involved in the nitrogen cycle. We mentioned before that most of the steps in the nitrogen cycle happen because of a microbe creating that step in the nitrogen cycle. Microorganisms are important to humans in making industrial chemicals, like we use microorganisms to make ethanol you put in your car and use it with gasoline to make your car run. We can use microorganisms to make acetone, other organic industrial chemicals. We can use microorganisms to make insecticides. We use microorganisms all the time in our fermented food, such as vinegar, cheese, bread, beer. All of these foods are made with microorganisms fermenting the food, causing the milk to change into cheese, the wine to change into vinegar, the grape juice to change into wine, or whatever hops change into beer. And then leavened bread actually uses fermentation to make the bread rise from the yeast. Otherwise, we'd only be eating unleavened bread, bread that did not rise. We use microorganisms to make products used in manufacturing, like cellulase. We use microorganisms to make antibiotics, like penicillin G is made by a microorganism. And then we use microorganisms to make other products used in in medicine, like human insulin. Today is made by a microbe. They put the human insulin gene in the bacteria and then grow up the bacteria to make the human insulin and then purify it and then sell it and give it to the diabetics. Any question about any of that? All right, let me talk a little bit about biotechnology and genetic engineering. Biotechnology is the use of microbes and other organisms to produce foods and products for human use. Biotechnology is centuries old. Like we use biotechnology to make kimchi and sauerkraut. Having the microbes make the kimchi and the sauerkraut. That is centuries old. Any question about any of that? So biotechnology is not something new. It's centuries old, and people have been doing it for at least centuries, probably close to 1,000 years, making wine. And actually, wine would be closer to at least 2,000 years, maybe 8,000 years. Genetic engineering is a new technique of biotechnology. And generally, it's what we think of when we talk about biotechnology, where you take genes from an organism, insert it into another organism, that's genetic engineering, and then make that other organism make a product of interest. Applications of genetic engineering include producing a variety of proteins from bacteria and fungi. Like we can make penicillin G, which really isn't a protein. I'm not sure what penicillin G is. It's an antibiotic. Put the gene into bacteria. Of course, there wouldn't be penicillin G. That comes naturally in a fungi. But you can put it into another organism and grow it up. Another example would be they've taken the human insulin gene out of the human and then put it into a bacteria and then grow up the bacteria to make human insulin, which is also not a protein. I'm not sure what insulin is. It's a hormone. You can use genetic engineering to engage in gene therapy. Like there are children born today which have defective genes in their immune system, so that these children, for example, cannot make either IgA or IgG, and you can take the correct human gene out of a human, put it in a microorganism, usually it's a virus, and then infect the cell with the virus and take the child's cells, their immune system cells, grow them up in vitro, infect them with the virus containing the correct gene, and then put those cells back into the child, and then they'll have functioning, normal immune cells that make IgA and IgG. And that is gene therapy. That's been done for over 20 years now. There are other applications of gene therapy, although I think they're still experimental, like they're experimenting putting the human insulin gene into some tissue in vitro and then growing the tissue up and then transplanting it into the human. This is still experimental. And
Yeah,
but
I was just gonna like talk about that just for a second. I'm type 1 diabetic and I'm so surprised that they're doing all those studies because I was told by my diabetes team that they're trying to like create like a fake pancreas that does exactly what you're saying so that we don't have to like stick ourselves anymore.
yes, like I said, it's all experimental with the insulin, but they have islets of Langerhans cells that they've put in the correct gene, and that would be gene therapy. And then, like I said, they've put these cells into patients, but it's still experimental. And I think the problem is that you will have to stick yourself every occasionally because you have to replace those islets of Langerhans cells. Like
Yeah,
the
that's what I
children
thought was crazy. Keep
go
going.
on.
I was like, my doctor was telling me about that. And I was thinking, because I use a pump and I have to replace it every like 2 days because the insulin goes bad. So, would like with this surgery, and I know you might not know because like it's getting tested now, but like, would you have to go in and reopen your pancreas to put like the new isolate cells in there and stuff? Or like, I don't know. I just feel like that would be
I
so.
don't know that question. I'm not a doctor and I'm not familiar with this experimental technique. Like I said, with the insulin, I know it's only experimental, but they are working on it. But with the children who are born with a defective immune system, they can put in the immune system cells that have the correct gene, and then their immune system cells are able to work, making like the IgA or IgG or IgM, whatever. And the trouble is, those cells only last for a few months. So every few months, I don't remember if it's every 3 months or every 4 months, the child has to go in and receive this therapy again, where they pull out the child's cells and then infect them with the correct gene and then put the cells back into the child. So every few months, the children get it. And the last I knew, there was like a 10-year-old child who she was now in her 20s. She had been living with the gene therapy for over 10 years. And that's at least 10 years old. So now she must be in her 30s. But like I said, um, with the gene therapy, you usually have to go in for more than one session. In fact, it's several sessions, 3 or 4 sessions in a year. I don't know what it would be with the insulin gene, but my guess is the islets of Langerhans cells would need to be replaced. They might be replaced much less often than the immune system cells, because immune system cells typically don't live that long. And that's why the child has to receive therapy every 3 or 4 months. And I don't know how long the islets of Langerhans cells last, but when they die, you would have to get more cells. All right, let's move on to genetic engineering. There's also genetically modified bacteria, like we have some bacteria which are given a gene to help protect the crops from insects so that the bacteria would kill the insect when the insect were to eat it. Or you can put in a gene in the bacteria that makes the bacteria make antifreeze, and then that keeps the plant from freezing. So those are some genetically modified bacteria. I gave you some examples of gene therapy, and then some examples of making products in bacteria and fungi. Any question about any of that? All right, let's move on. Microbes are important in human welfare, like microbial ecology. Bacteria, I've already mentioned, recycle nitrogen in the Earth's ecosystem, moving it from one form of nitrogen to another on the planet. Well, microbes are important in the recycling of carbon, the recycling of sulfur, the recycling of phosphorus on the planet. And it's true, microbes are involved in these chemical recycling on the planets, but they're not as important as microbes are in the nitrogen cycle, where almost every step in the nitrogen cycle happens because of a microbe, like in the carbon and the sulfur cycle. These atoms can come out of rocks just by water leaching it out of the rocks. So my point is that the microbes are important. They're not quite as important as the microbes in the nitrogen cycle, but you do need microbes to make the carbon cycle, the nitrogen cycle, the sulfur cycle, and the phosphorus cycle recycle the elements on the planet Earth. Then they can be used by plants and animals and other microbes. And then microbes are extremely important in nutrient recycling. The only reason why nutrients recycle on the planet is because microbes decompose the dead living matter, or what was once alive, and then make those nutrients available to other organisms on the planet. All of these are examples of microbial mediated biochemical cycles that support life on Earth. And these microbial biogeochemical cycles were first discovered by Dr. Winogradsky and Bajering, who showed the recycling of nutrients, specifically nitrogen and sulfur, in the ecosystem. And they discovered this in the 1880s to 1892. Any question about any of that? The reason why Winogradsky and his associate Bajering were able to discover this and other microbiologists were not was Winogradsky did not study an organism in its pure isolated form. Most microbiologists study a microorganism by purifying the species so there's only one species that they have, and then they study it. It's much easier to study if you purify the organism, because if you have multiple organisms in a test tube, and then the test tube gets a result, let's say it changes to yellow in color, you don't know which organism made the change in color. But if you have only one species in there, and it's a pure organism, and the test tube turns yellow, you know that that one species caused that test tube to turn yellow. Dr. Winogradsky, instead of studying pure organisms, he studied microbes living all together in the natural ecosystem. And that's why he was able to discover the nitrogen cycle and the sulfur cycle, as well as the other microbial mediated biogeochemical cycles. And that's because to get these cycles to run, you need lots of organisms. One species can only change one cycle of the nitrogen cycle, one step. So to get the entire nitrogen cycle to run, you need all the different species in the nitrogen cycle to operate. And that's why Winogradsky was able to study it. Any question about any of that? All right. Microbes are important to human welfare. Bioremediation literally means life endermity. We use bioremediation, meaning microbes, to help solve problems, like an environmental problem. Like these two gentlemen here are spraying microbes on this oil spill, and the microbes are digesting the oil, breaking it down into CO2 and water. We've already mentioned that microbes are important in sewage recycling, and it's the microbes in the water of the sewage that break down the sewage, breaking down the carbon organic matter in the sewage to CO2 and water. We can use microbes to treat pollutants, like bacteria can be sprayed on the oil to break it down. And then we can use microbes to also make the pollutant mercury less toxic. The bacteria do not break down the mercury. That's an element, so you can't break it down to anything further because it's an atom of mercury. But the bacteria can convert mercury from one form to another, making it less toxic form of mercury, like elemental mercury, which would just be the naked atom of mercury, is fairly toxic. And you put bacteria on it, it can make organic mercury, which is less toxic to humans and other organisms. Any question about any of that? So we can use microorganisms to engage in bioremediation, which, like I said, we do tend to spray oil spills with microbes to help break it down. But another thing is, of course, we've had so many oil spills on the planet Earth now. There are microbes on the planet which will grow naturally in the oil, breaking it down. So eventually the oil will be broken down by the natural microbes in the planet. We can use microbes to make biological insecticides. Some microbes are pathogenic to insects, and so they can be alternatives to chemical pesticides. One advantage of this is these biological insecticides are not as toxic to humans or organisms as chemical pesticides. So we can use these microbial pesticides to prevent insect damage to crops, as well as reduce the disease transmission among crops. An example is Bacillus thuringiensis. This bacteria is fatal to many insects, but it's harmless to humans and other animals, as well as plants. And there we're looking at, let me blow that up, Bacillus thuringiensis right there and there, maybe right there, but definitely there and there. And this is toxic to many insects when they eat it. And yet this is a package of Bacillus thuringiensis, and you can actually open this package and then put it on your plants to protect the plants from insects. My father actually did this back in the 1960s. That was a long time ago, right? My father's now dead, so obviously it's a long time ago. He put it on the apple trees to prevent the apples from getting worms. The trouble was my father was too lazy and he only put it on the apple tree one time. And you need to put it on about every 2 weeks, which is the same schedule. You have to use chemical insecticides. You have to spray the apples every 2 weeks, or else they can get worms. And then he was doing it in Western Oregon. If you know, there's a lot of rain in the fall of Western Oregon, and you have to put it on every time after it rains because the rain washes the Bacillus thuringiensis off of the apples. So he got it on the apples, and the worms were less than normal, but it wasn't worm-free. But like I said, he didn't do it correctly. He should have read the directions, and it told him to do it every 2 weeks, and then after every rainfall. And if he would have done that, there would have been no worms on the apples. And this is an organic way of growing apples, by the way. Any question about it?
So yeah, when that gets like sprayed on the apples, can we still eat it?
Yeah, Bacillus thuringiensis, I've already mentioned, is harmless to humans. It wouldn't be a bad idea to rinse off the apple just to rinse off the Bacillus thuringiensis, but if you get it, it's no big problem. Okay? All right. Any questions about microbes and their benefit to humans or their benefit to the planet Earth? If not, I'm going to change gears a little bit and start talking about the naming and classifying of microorganisms. This guy here began the scientific nomenclature, meaning naming all organisms, and he also began classifying all organisms, including microorganisms. Anybody know who this is? The guy who's the father of scientific naming or nomenclature, as well as the father of classifying organisms. You should have had that in Biology 160.
Linnaeus.
Yeah, Linnaeus, or Linné, if you take his, I think it's Swedish name. His Swedish name is something like Carl Linné. However, he liked Latin so much that he Latinized his name. And your textbook calls him Carl Linnaeus, but he actually called himself Carolus Linnaeus. So either Carl Linné, Carl Linnaeus, or Carolus Linnaeus, they all are the same person. And like I said, he just liked Latin so much that he Latinized his own name. But he was born Carl Linné, assuming that I'm pronouncing that correct. And I'm not Swedish, so I might not be getting the pronunciation correctly. But he established the scientific nomenclature as well as the first classifying of organisms. In his nomenclature, he set up each species or each organism so that the excuse me here, that the scientific name had two names, a genus name and the specific epithet name. So the species name, Homo sapiens, had the genus name Homo and the specific epithet sapiens, and you need two names to get the specific species Homo sapiens. Any question about any of that? This is called the scientific nomenclature or the binomial nomenclature because all names have two names to the species. Any question about any of that? Now, he did begin the classifying of all organisms as well. However, we don't use his classification system anymore because he classified all organisms into a plant or an animal. And as you know, there are more organisms than just plants or animals. For example, I think he classified microorganisms into plants. And microorganisms are not really a plant, they're not an animal, they are something separate, we'll just word it that way. Their own kingdom, or actually their own two domains. So we don't use his classification system, but we still use his nomenclature, meaning his naming system, where all species have two names, one for the genus and one for the specific epithet. and you need both to name the species. All scientific names are italicized, or if you're writing them out and you can't write in italicized, what you do is you underline it to mean that it is a scientific name, and italicized if you were to type it out. The genus name is always capitalized. The specific epithet is always in lowercase. The names are Latinized and they're used worldwide. So anywhere you go on the planet Earth, no matter what language the people speak, Homo sapien will be used by everyone to mean human. Any question about any of that? So it's used worldwide. The name may be descriptive or it may honor a scientist. Some examples: Homo sapien. Homo means, I think something like ape or person who walks on 2 feet, 2 legs. Sapien means wise, so wise man. Escherichia coli. Coli is descriptive. This is where we find it in the colon. You can also find it in the lower intestine, but colon is one area we find it. Escherichia is named after Dr. Esther, an early microbiologist. So that's honoring a scientist. Staphylococcus aureus is descriptive. Aureus means golden. The cells are golden yellow in color. Staphylococcus is a coccus or a round ball. Staphylo means the round balls growing in a cluster like grapes. That's what Staphylococcus means. Cluster like grapes, and of course the grapes are the shape of the cocci, the ball. Any question about any of that? All right. So when you first use a scientific name, you should spell out both the genus and the species name, specific epithet name, sorry, not the species name. You should spell out the entire species name. But after the first use, you may abbreviate the genus by just the capital letter of the genus, and then a period, and then spell out the specific epithet. So Staphylococcus aureus, after the first use, can be abbreviated as S. aureus. Escherichia coli can be abbreviated as E. coli. Both of these organisms are found on the human body. And I should mention something about E. coli. E. coli is so common, so commonly used, that people are much more likely to call it E. coli than Escherichia coli. Even among scientists, they will frequently call it E. coli the first time they use it because that name is so familiar, everybody knows what it is, even though they should properly spell out Escherichia coli the first time. But like I said, this is an exception. It's so commonly known that it is frequently called E. coli the first time it's used. And that's the only one that I'm aware of. Any question about any of that? All right, let's return to the classifying of microorganisms. Classifying of microorganisms follows the classification of all living organisms. And we have to look at all living things on the planet Earth. And then we classify all living things into 3 domains. I go backwards? Yep. The 3 domain system is also called the tree of life because the living organisms branch on a three-trunk tree, meaning there's one branch, which then branches into smaller branches, and this is called the tree of life. The three-domain system is based on genetic data, meaning the sequence of the DNA determine which domain you are in. All bacteria have a sequence of DNA which is more similar to another bacteria than to any organism outside the bacteria, which there's three domains: the bacteria, the archaea or archaebacteria, and then the eukarya. So if you're a E. coli, for example, your sequence of DNA will be much more similar to Staphylococcus aureus than to any life form in archaea, any organism in archaea, or any organism in eukarya. So the three-domain system is based on the genes, the sequence of the DNA. And if we ever find a new organism, they just sequence its DNA, and then it tells us which domain the organism falls into. Any question about any of that? So the important thing to realize is that the domain is classified. There's no argument about where an organism goes because you just sequence the DNA and then it tells you which of the three domains the organism goes into. If it's mostly similar to other eukaryotes, it belongs in the domain eukarya. If it's mostly similar to the archaea, then it belongs in the domain archaea. And if the sequence is mostly similar to the domain bacteria, it falls within the domain bacteria. Now, there are kingdoms, but you should realize the kingdom is not defined. There are different numbers of kingdoms. You could have 3 kingdoms, in which case they're the same as the domain. You can have 5 kingdoms, or 6 kingdoms, or 9 kingdoms, or 15 kingdoms, or 20 kingdoms, or higher numbers of kingdoms. Like I said, the number of kingdoms is not defined, not agreed on, how many number of kingdoms there are, and then what the definition of a kingdom is not defined. So there's arguments about whether a organism belongs in one kingdom or another. Like the kelp that washes up on the Oregon beach or the Washington beach, some people argue it belongs to kingdom Protista. Other kingdom argue it belongs to kingdom Plantae. And it really depends on how you, I guess, define your kingdoms and how many there are. We're going to talk about the 6-kingdom approach because it's the last kingdom approach that all scientists agreed on. And it's also an easy kingdom approach, and it's the one your textbook discusses, so we'll just talk about it. And that is for domain bacteria, there's only one kingdom, and it's the same as the domain bacteria. The same with the domain archaea, there's only one kingdom, so the kingdom and the domain are the same. So there's a domain bacteria and a kingdom bacteria, a domain archaea and a kingdom archaea. And then there's a domain eukarya, but it has 4 different kingdoms. Kingdom Plantae, the plants, kingdom Animalia, the animals, kingdom Fungi, the fungus, and then the kingdom Protista, the protists. Any question about any of that? Now, let me mention that when we split the eukarya into these 4 kingdoms here, that the kingdom Protista is unique in the sense that it is not a monophyletic group. the kingdom Protista is a eukaryote, which is not a plant, not an animal, not a fungi, meaning it's not a monophyletic group. And that was always known. The way we define Protista is a eukaryote that's not a plant, not an animal, not a fungi. If you have higher order kingdoms, you simply split the kingdom Protista from one kingdom into other kingdoms. And that would be the 9 kingdom approach and the 15 kingdom approach. I'm not sure if that's the 20 kingdom approach. One of the higher order kingdoms also splits the kingdom Plantae into 2 kingdoms. And I think it splits the bacteria and the archaea. The archaea are split into 3 kingdoms. I don't remember how many kingdoms the bacteria are split into. And we won't talk about these other kingdom approaches. Like I said, the number of kingdoms, scientists are in argument about onto how many different kingdoms there are. And this argument has been going on for over 10 years. It may be over 20 years now. And scientists can't agree on how many kingdoms there are. And then, of course, they don't agree on what the definition of a kingdom is. And I don't think anyone has a definition for what a kingdom is. Any question about any of that? You can then divide the kingdom into different categories. So let me see now. King Peter's son came over from Germany Soust. So K would be kingdom, P would be phyla, O would be order, C would be class, F would be family, G would be genus, and then S would be species. So you do divide the kingdom into different organisms. We're not going to talk about further dividing the classification of life. Any question about any of that? Obviously, microorganisms fall into each of these 3 domains. Bacteria, all of the members of this domain are a microbe. All of the archaea are microbes. Only some of the eukarya are microbes. Let me blow this up. The animals are not microbes, the fungi. Some of them are microbes. The yeast are microbes, but the mushrooms are obviously not microbes. The plants are not microbes, but the ciliates, the euglena, Plasmodium, Microspora, the Archozoa. The slime molds are not microscopic. The omocytes are, the amoeba are, the chromoista are. So some of these are microbes. All of the members of the, let me make sure that's right. All the members of the kingdom Protista are microbes. And then some of the fungi are microbes. Any question about any of that? All right, the 3-domain system has what your textbook calls a cell type. And there are 2 cell types. Some people call it a super kingdom. And the super kingdom actually is higher than the domain. So you all have all living things. If you have a super kingdom, that's the next one, separating the all living things into a prokaryote or a eukaryote. Our textbook calls it a cell type, and it says the domain bacteria have the cell type prokaryotes, meaning they are prokaryotic cells. The domain archaea have the cell type prokaryote, meaning they are all prokaryotic cells. The domain eukarya have the cell type eukaryotes or eukaryotic cells. Any question about any of that? I tend to call it a super kingdom because that's the way I learned it, but you could call it a cell type because that's the way your textbook has it. The point is that there are two of them, cell type, prokaryotic cells, and eukaryotic cells. When we're talking about the domain bacteria, their cell type is prokaryotes. If they have a cell wall, they all have peptidoglycan in their cell wall. This is a characteristic of domain bacteria. If their bacteria has a cell wall, you can find peptidoglycan in the cell wall. The domain bacteria mainly multiply by binary fission. They can obtain their energy from organic chemicals, from inorganic chemicals, or from photosynthesis. Most of the domain bacteria are free-living decomposers or primary producers, but some of the bacteria are parasitic and some of them are pathogenic, meaning some of them do cause disease. Any question about any of that? The domain archaea, all of the cells or species are prokaryotic. Do the domain bacteria have peptidoglycan in their cell walls? Anybody know?
No.
They do not. You are correct. The domain archaea do not have peptidoglycan in their cell wall. So peptidoglycan, if it's found, that means it is from the domain bacteria because they're the only domain that has peptidoglycan in their cell wall. Some of the domain archaea are found in extreme environments, including methanogens, found in areas where there's methane. Some of them are found in extreme halophiles, that's high salt concentrations, like we find archaea in the Dead Sea between Jordan and Palestine, because they are extreme halophiles. And then we can find extreme thermophiles as archaea. These are organisms that live at high temperatures. But this is where we only first found the archaea, in these extreme environments. When we look further, we find archaea in all of the Earth's biomes. They are actually a significant portion of the plankton in the ocean, and they are a significant portion of the microbes in the soil, not bacteria, the soil prokaryotic life. So they're a significant portion of the organisms in soil. Now, I mentioned that the domain bacteria, some of them are pathogenic, some of them do cause disease. How about the domain archaea? Are any of them pathogenic? Anyone know? It's either a yes or no question, so you got to.
No.
You're correct. The domain archaea, none of them are pathogenic. We know of no pathogens in the domain archaea. So after this class, we will not be studying much about the domain archaea. And if ever I'm talking about them, I'll specifically state these are the archaea, because we don't talk about them in general. We talk about the domain bacteria in general because they are the microbes that cause disease. And the only reason why we're doing that is because this class is focusing in on the microbes that cause human disease. It's not because the domain archaea are not important or that I feel they are uninteresting. It's just that none of them cause disease, and the focus of this class will be on disease causing organisms. So because of that, after this lesson, we will hardly ever talk about the domain archaea. Any question about any of that? All right.
Did you say that there was no peptoglycin in it, right?
There's no peptoglycan in the domain archaea, in their cell wall. If there's peptoglycan in their cell wall, it's a bacteria. All right. Let's move on to the eukarya, the third domain. This domain we break into four kingdoms: the kingdom Plantae, the kingdom Animalia, the kingdom Fungi, and the kingdom Protista. All of these kingdoms, or all of the eukarya, lack peptidoglycan. Let's talk a little bit more about the kingdom Fungi. Most of them have cell walls. You don't need to know that they have chitin in their cell walls, but they have cell walls. They use organic chemicals for energy. That should be familiar to you because humans use organic chemicals for energy. The main chemical that fungi and humans use is, of course, glucose. The kingdom fungi includes the multicellular molds and the multicellular mushrooms, which consist of masses of mycelia, shown here. But each of these is a hypha. But the kingdom fungi also includes the unicellular yeast. So the yeasts are microorganisms, and they make up a significant portion of the fungi. There's also the molds, which are most of the fungi, or at least the plurality of the fungi. And then there's mushrooms. Most of the fungi are decomposers, but some of the fungi are pathogens. Any question about any of that? All right, let's move on to the kingdom Protista. The Protista can be separated into three groups: the algae, or the plant-like protists, the protozoa, or the animal-like protists, and then the fungus-like protists. The algae have cellulose in their cell walls, much like plant cells. They do engage in photosynthesis while they're green, although red algae is also photosynthetic, but it doesn't have the color green because it doesn't have the chlorophyll as its pigment. It has a red pigment. The algae all produce molecular oxygen, as well as organic compounds like glucose. And that's it for the algae, the plant-like protists. We won't talk much about them because the plant-like protists do not cause human disease. The protozoa include the amoeba, the paramecium, euglena. They all absorb or ingest organic chemicals like glucose. They may be motile, and if they are motile, they can move by pseudopods, by cilia, or flagella. And this separates out the eukarya from the bacteria and the archaea, because the bacteria and the archaea, if they move, they only move because of flagella. That's the only thing prokaryotes have to move with. Prokaryotes do not have pseudopods. They do not have cilia. The protozoa, some of them are parasites, so some of them can cause disease, like amoebic dysentery. Forgot the name of that one now. It causes sleeping sickness. It's a paramecium. Euglena is not a pathogen. It's not a protist. It's not a parasite. Euglena is a free-living photosynthetic protozoa, which doesn't have to be photosynthetic. It can consume glucose or ingest glucose and then live that way, in which case it would be acting like an animal consuming its food. And then the fungus-like protist, there's only one I'm going to mention, and that is the potato blight. The potato disease, which actually killed off the potatoes in Ireland, which brought a bunch of Irish to the United States. Potato blight is a fungus-like protist. Any question about any of that? The fungus-like protists do not cause human disease, so we won't talk about them further. We will talk a little bit about the protozoa in the future, like malaria is caused by a protozoa. And it obviously causes human disease. Any questions about any of that? All right, let's talk a little bit more about the eukaryotes. We can separate them into the kingdom Plantae. You can separate the plants into mosses, ferns, conifers, and flowering plants. All of the plants are multicellular. Let's move on to the animals. You can split the animals into the sponges, the worms, or helminths, scientific name, the insects, and the vertebrates. Humans are a vertebra. All of the animals are multicellular. There is one category of the animals we will talk about in this class, and that's because the worms, the helminths, some of them can be infectious, where the disease can spread from one person to another by passing the parasite or the eggs of the parasite. And we will talk a little bit about the worms because they do cause disease. All of the others we won't talk about because they do not cause, well, it's not entirely true. Insects can cause disease if they infect you with something, but not directly. The insect does not directly cause disease. Any question about any of that? All right, that's it for the kingdom Eukarya. I've already mentioned that a microorganism is used interchangeably with the term microbe. And what we mean by a microbe is a bacteria, an archaea, a virus, and a prion. Even though viruses and prions are exceptions, they are not organisms. They do not have cells. Certain of the eukarya are microorganisms as well, like the protozoa. All members of the protozoa are microbes. Some of the molds are microbes. All of the yeasts are microbes. Some of the algae are microbes. If you can see it, it is not a microscopic algae, but if it's single-celled or a small number of multicellular algae, and you can't see it without a microscope, then it is obviously a microbe. And then some of the helminths are also microbes. But wait a minute, the worms aren't microscopic, right? Most of the worms are quite big, at least the adults. The helminths are multicellular animals, in which case most of the adults, not all of the adults, but most of the adults are not microscopic. But we do consider helminths a microorganisms and something that biologists study for two reasons. One, some of the worms can cause disease, and microorganisms study infectious diseases, including the ones caused by infectious worm disease. And then the second reason why microbiologists study them is that usually the infective form of the worm is microscopic. So the infectious form of the worm is the stage of the worm that first infects a patient. And usually that is microscopic. It may be just the fertilized egg. It could be a microorganism that burrows into the skin of the patient. So some of the multicellular worms are infectious human pathogens or parasites, but not all of the helminths, not all of the worms are parasitic. Many are free living. For example, the annelids, an earthworm, all of that, I don't know if it's family or phyla, are not human pathogens, meaning all of the earthworms do not cause disease. But some of the nematodes or roundworms can cause disease, infectious disease, and then some of the flatworms can cause infectious disease in humans. And I think all the flatworms cause disease in one animal or another, just not all of them cause disease in humans. So the flatworms, that's not true. There are some free-living flatworms as well. So the helminths are not strictly microorganisms. Many of them, at least the adult form of the worm, is not microscopic. However, the stage which infects the patient is usually microscopic. And some of the life cycle stages will be microscopic. For example, with what is that called? The ringworm? Just say beef tapeworm. The infective stage in a human is the fertilized egg, which is microscopic. Any question about any of that? All right, any questions about anything? If not, I'll see you at 6:30 for the lab. Bye.